Hey everyone, and warmly welcome to this explainer. Today, we're taking a really clean, highly structured approach to one of the absolute most foundational topics in your medical studies, the Krebs cycle, or, you might call it the tricarboxylic acid cycle, or the citric acid cycle. This incredibly elegant biochemical pathway was mapped out by Hans Krebs, and I know, believe me, I know, that metabolism can sometimes feel completely overwhelming. But take a deep breath. We're going to walk through this together with a soft, reassuring, and completely organized study guide, so you can walk into your exams feeling confident and totally prepared.
Okay, let's dive right into this. Here is our syllabus for today. First, welcome to the mitochondria. Second, the Krebs cycle journey. Third, a midpoint knowledge check. Fourth, clinical pathologies and mutations. Fifth, enzymes and regulators. And finally, high-yield takeaways.
Section 1. Welcome to the mitochondria, setting the scene.
So before the main cycle even starts, we have to set the stage. Glycolysis has already done the heavy lifting of converting a single 6-carbon glucose molecule into two 3-carbon pyruvates out in the cytoplasm. Now, those pyruvates physically enter the mitochondria. They cross the outer and inner membranes to reach the innermost compartment, which is the mitochondrial matrix. It's really crucial to picture this spatial layout. The matrix is where the Krebs cycle actually happens, while that highly folded inner membrane, the cristae, is where the electron transport chain sits. Right here in the matrix, a vital transition step occurs. We add coenzyme A to our pyruvate, generate an NADH, and pop off a CO2 to form acetyl-CoA. And that acetyl-CoA, that's our golden ticket. It's the absolute requirement to begin our cycle.
Let's move to Section 2, the Krebs cycle journey, the sequential flow.
To ease the cognitive load here, we're going to anchor this entire pathway with a super simple high-yield mnemonic. Citrate is Krebs' starting substrate for making oxaloacetate. The cycle officially kicks off when our 2-carbon golden ticket, acetyl-CoA, fuses with a 4-carbon molecule called oxaloacetate. Simple math, right? 2 plus 4 equals 6, and that creates our 6-carbon starting molecule, citrate. Getting this sequence memorized right out of the gate is honestly the best way to just massively reduce your study stress.
Using that mnemonic, we can effortlessly track the whole journey. Citrate converts to isocitrate, isocitrate becomes alpha-ketoglutarate, then we move to succinyl-CoA, which turns into succinate. From there, we go to fumarate, then malate, and finally, we end up right back where we started at oxaloacetate, completely ready to fuse with a brand new acetyl-CoA and fire up the whole process again. It's just a beautiful loop.
Section 3, midpoint knowledge check. A brief review.
We all know active recall is a scientifically proven study technique, and it's absolutely vital for board prep. So let's pause for a second. Here's your question. Which Krebs cycle enzyme is also known as complex 2 in the electron transport chain and is uniquely anchored to the inner mitochondrial membrane? Take just a brief moment to lock in your answer. Did you get it? The answer is succinate dehydrogenase. This incredibly special enzyme sits right on the inner mitochondrial membrane, the cristae. During the cycle, its job is converting succinate to fumarate, and crucially, it generates FADH2 in the process. This is a massive board exam crossover fact because it physically links the Krebs cycle directly to the electron transport chain.
Section 4. Clinical pathologies and mutations. Real-world consequences.
Now, what's really interesting about this is how we can bridge dry biochemistry straight to the clinic. First up, aconitis. This enzyme can be fatally inhibited by fluoroacetate. That's a chemical found in rat poison that actually mimics acetyl-CoA. Next is alpha-ketoglutarate dehydrogenase. A very specific mutation involving NADPH here produces a rogue byproduct called 2-hydroxyglutarate. This byproduct actually halts gene regulation, leading to deadly brain tumors like glioblastoma. Then, succinate dehydrogenase. Mutations here can cause a pheochromocytoma in the adrenal medulla that dumps massive amounts of epinephrine into the body, triggering a severe hypertensive crisis. And finally, fumarase. A simple deficiency here can lead to leiomyomas. Those are benign, smooth muscle tumors, commonly seen as fibroids in the uterus or the kidneys. It's literally astounding how a tiny, microscopic metabolic misstep can lead to such profound clinical pathologies.
Moving right along to Section 5, enzymes and regulatory inhibitors, metabolic signals.
So the crucial point for exams is distinguishing between how energy is actually made. During the cycle itself, the enzyme succinyl-CoA synthetase releases coenzyme A. That release creates enough potential energy to convert GDP to GTP. Then an ADP molecule essentially comes along and pickpockets that phosphate to become ATP directly. That process is called substrate-level phosphorylation. Conversely,
oxidative phosphorylation happens much later. That's when the NADH and FADH2 we generate in the cycle are transported over to the electron transport chain to mass produce HEP.
Let's look at how the cycle regulates itself. You'll notice a clear pattern. When the cell's loaded with high energy, marked by abundant ATP and NADH, the key enzymes, which are citrate synthase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase, are inhibited. The body's essentially saying, hey, we have enough energy, stop producing more. On the flip side, low-energy markers like ADP stimulate the cycle. But look closely at calcium. Calcium acts as a powerful stimulator for both isocitrate and alpha-ketoglutarate dehydrogenase. Why? Well, in muscle tissue, calcium is the trigger for contractions, but those muscles absolutely need ATP to detach myosin from actin so they can form cross-bridges. So calcium is signaling the Krebs cycle to speed up, ensuring the muscles have the ATP they desperately need to function. It's just brilliant.
Rounding out the enzymes here, we can note which reactions are reversible, like aconitase, fumarase, and malate dehydrogenase. But listen, the major high-yield trick for exam day is the word dehydrogenase. Anytime you hear dehydrogenase, whether we're talking isocitrate, alpha-ketoglutarate, succinate, or malate, you should automatically, instantly know that a coenzyme is involved. You're either converting NAD+ to NADH, or, in the very special case of succinate dehydrogenase, FAD to FADH2.
Section 6. High-yield takeaways. A final review.
If there is one number you burn into your brain for net calculations, it's the number 2. Remember, because one single glucose molecule splits into 2 pyruvates way back during glycolysis, the Krebs cycle actually has to complete 2 full turns to process that single original molecule of glucose.
And from those 2 full turns, these are your exact, non-negotiable net products. You generate 4 CO2 molecules via decarboxylation, you generate 6 NADH molecules, and 2 FADH2 molecules. Finally, you generate exactly 2 ATP via that substrate-level phosphorylation we talked about. Now, those 6 NADHs and 2 FADH2s are fully loaded with hydride ions, and they are perfectly primed to head straight over to the electron transport chain for massive oxidative phosphorylation.
And this brilliantly illustrates the true beauty of studying biochemistry. It's really not just rote memorization. As we saw with the clinical mutations that lead to severe tumors or hypertensive crises, understanding how something so incredibly microscopic, something you might be tempted to dismiss as, oh, this is just metabolism, has an absolute systemic and amazing effect on the entire human body.
You now possess a deeply structured, truly clinical understanding of the Krebs cycle. So I leave you with this thought as you go back to hitting the books. How will you apply this foundational biochemical knowledge to actually diagnose and treat complex metabolic disorders in your future patients? Keep this structure in mind, trust in the hard work you're putting in.